Ribonucleic acid, or RNA. RNA, like DNA, is a long, unbranched polymer consisting of nucleotides joined by phosphodiester bonds. The number of nucleotides in an RNA molecule ranges from as few as 73 to as many as thousands. The primary structure of RNA differs from that of DNA in two ways. As we saw in chapter 13.2, the sugar unit in RNA is ribose rather than deoxyribose, which is found in DNA. The other difference is that RNA contains the base uracil instead of thymine. So we have still our C, which is going to be paired up with our G, but now instead of A being paired up with our T, we're going to have U being paired up with our T. The secondary structure of RNA is also different from that of DNA. RNA molecules are single-stranded, except for in some viruses, and consequently, an RNA molecule need not contain complementary base ratios of 1 to 1. In general, however, RNA molecules are highly structured and contain stem loop regions where the stem portion consists of bases, the hydrogen bond, with the corresponding bases in the same RNA strand. And you can see that concept right here in this figure where we have this loop, right? It's being looped around. And you can see the A's hydrogen bonding with the U's and the C's hydrogen bonding with the G's. Now, the proportion of the helical regions varies over a wide range depending on the kind of RNA that you're looking at, but a value of around 50% is typical. RNA is distributed throughout cells. It's present in the nucleus, it's present in the cytoplasm, and in mitochondria. Cells contain three main kinds of RNA. We have messenger RNA, or mRNA, ribosomal RNA, or rRNA, and transfer RNA, or tRNA. And each of these kinds of RNA performs an important function in protein synthesis. Messenger RNA, or our mRNA, functions as a carrier of genetic information from the DNA of the cell nucleus directly to the cytoplasm, where protein synthesis takes place. The bases of mRNA are in a sequence that is complementary to the base sequence of one of the strands of the nuclear DNA. In contrast to DNA, which remains intact and unchanged throughout the life of the cell, mRNA has a very short lifetime. It's usually less than an hour. It's synthesized as needed and then rapidly degraded to its constituent nucleotides. In 2009, Ada Yonath, that you see here, won a Nobel Prize in chemistry for studies of structure and function of the ribosome. The function of ribosomes at the sites of protein synthesis is discussed later on in section 13.7, but briefly, ribosomes are large complexes that are composed of about 65% ribosomal RNA, or again, our rRNA, and they're about 35% protein. Overall, our RNA constitutes about 80 to 85% of the total RNA in a cell. Because of their small size, a number of tRNAs have been studied extensively. You see the figure that's shown on this slide, figure 13.20. It shows representations of the secondary and tertiary structures of a typical tRNA. This tRNA molecule, like all others, has regions where there is hydrogen bonding between complementary bases. And you can see those kind of regions like here and here, for example. And then it has the loops where there's no hydrogen bonding, and we see a loop here and here and here where we don't have any hydrogen bonding. Two regions of tRNA molecules have important functions during protein synthesis. One of these regions, designated the anticodon, enables tRNA to bind to mRNA during protein synthesis. The second important site is the 3' end of the molecule, which binds to an amino acid and transports it to the site of protein synthesis. And so you can see how we have our anticodon site here. And there's another one here. And then this is a very simplified version of our tRNA here. And then we have our amino acid attachment site up here at the 3' end. And you can see here in this diagram, which shows more of the three-dimensional shape of the molecule. And then we have the very simplified version right here. Each amino acid is joined to the 3' end of its specific tRNA by an ester bond that forms between the carboxyl group of the amino acid in the 3' hydroxy group of the ribose. And you can see that right here. Here's the carboxyl end or carboxy end of our amino acid, and here's the 3' end of our ribose. The reaction is catalyzed by an enzyme that matches tRNA molecules to their proper amino acids. These enzymes, which are very specific for both the structure of the amino acid and the tRNA, rarely cause a bond to form between an amino acid and the wrong tRNA. When a tRNA molecule is attached to its specific amino acid, it's said to be activated because it's ready to participate in protein synthesis. The figure shown in the yellow box shows the structure of an activated tRNA.